Welcome to Professor Parkinson's lab. My name is Ehsan Yazdanpanah Moghadam. I'm a PhD student in mechanical engineering of Concordia University. I've developed micro devices to study the cell's activities in neurodegenerative diseases in a more precise, inexpensive, and controllable methods.
Hi, my name is Sylvie Ouellette. I'm a PhD candidate in chemistry and biochemistry at the Loyal campus at Concordia University.
Hi, my name is Hamid Ebrahimi at Orimi, and I'm a PhD candidate at Mechanical Engineering Department of Concordia University.
This is a micro device. It consists of PDMS, is a type of cell-compatible polymer, and it's bonded to the glass slide. As you can see, there are some microchannel inside a PDMS that are shown with the red color. Actually, my project has two parts, biology and engineering. In the engineering part, I've designed micro devices in related software to predict the cell's fluid flow behaviors within the device. Based on the results, I fabricate my devices. Let's go to show how to fabricate microfluidic devices. Here is clean room, where I fabricate my devices. Using a microfabrication process, I make the mold over the silicone wafer, the device mold. It's a silicone wafer, and each of them is a device mold. After pouring the PDMS over the silicone wafer and cure it in the oven, I'm gonna get something like this. It's a pure PDMS and it's silicone wafer. When I peel off the PDMS here over the silicone wafer, you see the features of the device on a PDMS. You see like some channels, and then bonded on a glass slide. Here is the biological section of our lab. After testing the device under the cell hood, I load the immune cells of the brain and a type of proteins that play an essential role in progressing neurodegenerative diseases. And next step, I grow or culture them in an incubator. By tracking the cell's activity using microscope and live imaging, I've quantified the cell's activities toward the protein. Due to the micro-scale device dimensions, I require the minimal samples and reagent in experiment, which makes this method cheaper than the conventional approach. This device is capable of integrating microsensors to monitor the cell's activities precisely and continuously. This device also integrating with micropump or syringe pump to make a flow within the device. All these advantages of this microfluidic device can make it as alternative platform for a study in neurodegenerative diseases. Thank you. If you have a question, you can ask.
Thank you, Ehsan. If I may start with the first question, what type of cells are you working with, and is that from a tissue culture bank, or do you load in cells from patients, for instance? Is this something that can be used for individual? For instance, if I had a biopsy of my own brain cells, can you grow them and upload them onto your device for studying my own brain?
Actually, the neuron cells and a type of tau protein, as I mentioned, is, let's say, tau proteins and microglia cells. All these three elements play an essential role for progressing Alzheimer's disease. The cells I'm using is microglia cell line, BV2, and tau protein. I don't use neuron cells. And so it's like, because neuron cells produce the tau protein, and so in my experiment, at least at this level I'm working, I don't use the neuron cells, I remove it, and like load the tau protein directly in the device. And it's like expose it to microglia cells.
Thank you, Ehsan, for such a nice presentation and the project you're working on. Before going to deep in more details about your project, can you give us some commercial and practical example of micro devices?
If I want to give you some commercial examples, I can say home pregnancy test. It's a type of microfluidic device, or you can say a test kits for diagnosing COVID-19. And recently I read a journal that use a small chip. It's like a micro device, is attached to your back, and that analyze your your health using the suite.
I'm very interested in the cell culture aspect of your work. So from what I understand, you will take cells from your tissue culture, you will seed them onto your micro device, and then you use, is it microfluidics to keep them alive and fed?
Yeah.
Can you explain a bit more on that? And can you tell us, how do you make sure that they don't grow too much?
So first of all, as you know, if you want to keep alive the cells, we have to provide nutritious for the cells. It's a first step. In microfluidic, I designed some reservoir at the end of each channels to like supply nutritious to the cell. So in this way, I can keep alive the cells. And you asked me about overproliferation. When you load the cells inside a device, the cells it's not confluent inside the device. It's like the cells don't cover hold the surface. After 2 or 4 days, the cells get confluent. It means cover hold the surface. And when the cells cover hold the surface, I'm gonna start my experiment. And to check it, to check the overproliferation, I'm gonna use the microscope each 12 hours.
Okay, Ehsan, I'm interested to know about during your presentation, I was watching your device, which you are studying your Alzheimer's disease, and it was a very tiny device. It's like a microenvironment. Why we need such a tiny environment? Why not bigger? As I know, the bigger, the easier to fabricate. Can you just give us some hints about such a micro fabrication?
Yeah. As you know, Hamid, when you go to the centimeter scale, you're going to have more area. It means you have to monitor cells you have to monitor area in like bigger area. But when we work on the micro scale, you can focus more on the cells because you have less area to analyze, to monitor them. And so another reason, these micro devices have many options. For example, sometimes you want to trap a single cell. You can do it with microfluidic, and you want to array the cells side by side. For example, you want to array the neuron cells side by side and focus just on one single cell. And sometimes you want to stimulate just single cell. So you can integrate a nano-electrical element under the under the device and stimulate the cells. When you go to centimeter scale, for example, Petri dish, you don't have those options.
And you said micro devices are more affordable. Is that the only advantage that they have compared to conventional method, or there are some others?
I mentioned some advantages I explained for you, and I can say, as I said, it's so cheap because it's a cost when you go to the mass production. It's going to be the price of the chip is around $2, between $2 or $1. And on the other hand, sometimes you want to integrate micro syringe form. You want to as I mentioned, integrate microsensors. So why we are using the micropump or syringe pump, because actually sometimes you want to see the cell's activities in exposed to fluid flow inside the device. So because actually, as you know, some of our cells in device in exposed to fluid flow is like blood flow. And so it's it's some advantages I can say about why we are going to microscales or microfluidic devices.
Great, thank you. I'm curious about your design. During your presentation, I was watching some micro channels, so many channels, and can you give us some, could you please let us know why we need so many channels? Are they mimicking biological things or mechanical things? Yeah, that's my question.
Okay, Hamid. This cells like to migrate the cell-free area, and always it's migrates or move to cell-free area. In biology, it's talk about migration assays, conventional migration assays that are used to quantify the migration of the cells or cells activities. One of them is a scratching assay. I'm sure you and Sylvie know it. It's for example, it's Petri dish, and cells hold the surface of the Petri dish. You assume it. And so it's a pipette. They make a scratch over the surface. When you scratch over the surface, you're gonna make a cell-free area between the cells. It's like a line between cells. When you make the cell-free area among the cells start to migrate to cover the gap. So and then you can quantify the migration of the cells. I'm using these methods, but not it's the same way. I forgot to say this method is like a scratching assay. When you scratch or scratch over the surface, we are damaging cells at the edge. But I'm going to model this this method, but without damaging cells at the edge. What I'm gonna do in my project, I make the cell-free area, but without damaged cells. For example, I'm gonna give you an example. If you assume if for example, if you have two channels, it's like plus shape. It's when you look at my device, there are two perpendicular channels. And you see the cells, you see at the beginning, I allow the cells to cover whole the surface. Let's say when the cells get confluent. When the cells confluent, I'm gonna make the flow from here to here. It's like vertical channel. It's like trypsin flow. Trypsin flow is a liquid can use to remove the surface from the substrate. Okay, so I'm gonna use the trypsin flow from here to here. It's like it just guide the trypsin flow move in vertical channel. When you remove it, I expect the cells are removed from the substrate. You see just the cells left inside channels, here and here. In this way, I make the cell-free area between cells. And then cell migrate to like the cell-free area. In this way, I can quantify the cell migration. So what you saw in the device, I made some this type of interactions inside the device. So you can see like some perpendicular channels, vertical channels, perpendicular channels. You see like 3 or 4 interaction
I see.
channels here. It's like a whole idea behind of my device.
Nice idea. Yeah, thanks. I was wondering about the silicone mold fabrication. You went to the clean room and you start to work with some equipments. Can you give us how you're fabricating the device? Why you need clean room? Why not, I mean, in the lab environment, why you cannot fabricate?
Let's start for clean room. As you know, if you like, for example, here, you can find a lot of particle and dust. But when I want to fabricate my devices, I go to clean room because the clean room minimize the dust and particles in the air. Why? Because I'm working with the micro scale. If I don't work in the clean room, maybe a small particle or dust come and block my channel. So that's why I'm working in clean room. You asked me about the device modes. Let me show you. Here is silicone wafer, okay? It's a silicone wafer. Here is like there are some device modes, but you assume it's like just silicone wafer, okay? Without any modes. At the beginning, I have silicone wafer and pour the photoresist over the silicone wafer. This type of photoresist, it's cured and exposed to the UV light. I'm using mask something like this. You see here, there is a like black color and white color. The white color allow the UV light is passed from transparent area. So here I'm gonna, it's like silicone wafer. Over the silicone wafer, I have photoresist. If it's exposed to UV light, they can be cured and attach the surface. And then I'm gonna put the mask on the top of them, okay? When I expose it on the UV light, the UV light just pass from like transparent area, past the transparent area and cure my desired design. And so, but that area in the like black color is not cured. When you wash it with the solver, just you see just plus like shape of channel is left. You see?
And then after having the mold, you have to pour it with PDMS.
Exactly. As I explained in the clean room, when I like make the mold, I pour the PDMS over it and put it in incubator to be incured. When you incure, I'm gonna peel off the PDMS over the over the silicone wafer. When you peel off the silicone wafer, you see the features of the device mold over the PDMS. I'm gonna attach it to silicone. It's like glass slide. It's like, because actually my device needs a substrate. I'm using like a glass slide as a substrate.
And is there any reason to choose the PDMS? Why not other material?
Yeah, actually, the PDMS is biocompatible. And so I can culture cell in it without any problem. And so the PDMS is, can I have it? This PDMS is, you see, is transparent. And it's transparent and it can monitor the cells under the microscope, because inside the channel there are cells. And when you put it under microscope, it's transparent and it can see the cell's activities. And it's a cheap, it's low photoresist. And you see it's deformable. You look, it's deformable. And so sometimes I saw use the hydraulic and use these characteristics of the PDMS and use the hydraulic valves to like open and block the channel.
Thank you, some very clear answers. Let's say you have this Alzheimer microchip for treatment or diagnosis. I guess the plan for such a device you developed is to study the treatment application. Am I right?
No, it's not treatment, but I can use it like different drugs. I didn't open that area is related to drugs, but actually I can use it drug. For example, the work I'm doing is like, as I mentioned, is quantify the immune cells of the brain toward the tau protein. Or so you can like, let's say I'm going to make the microfluidic migration assay. So it's it's a final target of each migration assay is like check the different drugs. And you see this drug, for example, this drug has an effect of inhibitor or not. But I didn't open that area. Just I'm working on the design of this device. Is like, is it working? And I'm focused on like immune cells of the brain and tau protein. Maybe next students or like use it. And when you like work on it and develop it and make it conventional approach, a biologist can use it. It's like biology can use it. It's like a small chip and to quantify the migration of the cells.
Okay, great. Last but not least, what are the adding value and the contribution of your work in the field?
Yeah. Actually, Hamid, as I mentioned, I have two parts in my project, engineering and biology. Let's start to like novelty and contribution in engineering, in biomedical engineering. As I mentioned, it's like to make the migration assay, they try to make the cell-free area. And so, for example, as I said, like a scratching assay. When you look at the literature review, you see some people try to make the microfluidic migration assay, but they use a hydraulic valve, they use a pneumatic valve, they use like a micro syringe pump in the device to make the like flow inside the device. I make the flow inside the device by using gravity without anything. Just it means as long as you have this chip, you can quantify the migration. Let's start to explain how I make the flow just with gravity. For example, we have two surfaces, and each of here is topper from this one. When you make the flow here, I expect the water comes up or make the flow from up to down. It means the flow, the water flow from higher potential to lower potential. I'm doing same thing in my device. I make a trypsin flow by making different altitude surface, make a different altitude in my device to make the trypsin flow. It means just with help of the gravity, without any pre-products. It's a novelty. It's a novelty of my device in engineering part. If you go to biology part, actually in Alzheimer is there are three types isotau protein. As I say, tau protein, and it's tau protein is one of the reason of the Alzheimer disease. I don't go to the details. There are three types of tau proteins in the brain, and it's still, it's not fully understood. Microglia prefer to engulf or remove which type of tau protein, or let's say which type of tau protein can stimulate more microglia cells at a migration assay. And is there any bias for microglia cells in the brain to remove some type of tau proteins at the beginning and then go to remove others? And yeah, we are going to quantify and quantify this part of biology.
Thank you, Ehsan, for sharing this amazing project for us, and I believe it will help a researcher for further development in future. Good luck for the rest. Thanks.
Thank you, Hamid.
Bye, everyone.
Talk later.
Bye.
Bye. You.